Okay, we're going to talk about gene expression. In the last chapter, we talked about replication, DNA replication. Here we're going to talk about transcription and translation. So just to kind of give you a little bit of history here, we have the Beetle and Tatum experiment where they looked at mold spores and they demonstrated that one gene codes for one protein. In this case, it was an enzyme. So they took Canidia, which is a fungal spore. They um, put X-rays or UV radiation to cause a mutation. And what we found was that the number one right here, number one, we had growth on the bra and no induced nutritional mutation. Then we had this one right here. Okay. So this one here was mutated. So with that mutation, they grew, but when they put them on a medium with minimal materials in it, they could not survive. So there was an enzyme that was mutated that did not allow them to grow with the minimal nutrition in the uh, test tube. Okay. So after refining the one gene, that should say one protein, sorry. Hemoglobin we know hemoglobin has a quaternary structure, and we have two polypeptide chains that come together to make up a protein. All right. In fact, we've got the alpha and the beta, and we actually have two of each, so it's four total. This mutated gene only affects the polypeptide chain. Therefore, it is now noted that one gene, one polypeptide. We have our genetic code. We know that there are 20 amino acids. We know there are 4 nucleotides. So the question is, how can only 4 nucleotides code for 20 different amino acids? So if we were to read DNA one base at a time, you could only get 4 different amino acids. If we read them 2 bases at a time, we could only get 16. So we still don't have all the amino acids covered. However, if we read the DNA 3 bases at a time, you get 64 different amino acids. So we have plenty of combinations here to more than cover 20. All right, so we have our codon. Our codon is a triplet code. Our genetic code is what we call degenerative. It's degenerate because more than one codon codes for one amino acid. This code is universal. It is found, all the organisms on our planet go by this code. Hence, another support for evolution. So most organisms, DNA uses the same code, like I said, strong evolutionary relationships. Mammalian mitochondria and chloroplast DNA do differ slightly. So when we make a copy of messenger RNA from DNA, that's what we're going to do for transcription. So when we have our transcription, the first thing we need to do is this TATA box is really kind of important. So this being the gene that we want, all right, the template strand means that's the one we're gonna base pair with. That's what we're gonna read for our protein. So first of all, our transcription factor binds to the TATA box. That's kind of like, okay, we're going. Oh, excuse me. Okay. So we then are going to have RNA polymerase come in once all these transcription factors, you can see them all here, bind. It's going to unzip, it's going to open up a bubble so we can start copying our DNA. So here's that transcription bubble. We're going to unwind the DNA. We're going to open it up, and then we're going to go ahead and we're going to copy our messenger RNA. So for termination of transcription in prokaryotes, transcription continues until a termination sequence. In eukaryotes, it will continue until we have a sequence of AAU, AAA, often referred to as polyadenylation. Proteins in the nucleus recognize this sequence and the synthesizing messenger RNA. At this point downstream, these proteins will cut the pre-mRNA free, which releases the RNA polymerase. So, transcription termination. So again, eukaryotes have that polyadenylation sequence. So we can see we have our polyase signal here. We have our RNA polymerase. When it gets to that sequence, it is going to, the RNA polymerase will fall off. And notice our messenger RNA is released. Okay. Oh, that must be it. All right. So you can see here, we're going to talk about the poly A and the cap here in what's referred to as the mature messenger RNA. But for these two sections, they're just basically going over how what is transcription. All right. So that was kind of short and sweet. Again, any questions? Just let me know in class or by email. All right. Have a nice evening. Bye.